The overall goal of this procedure is to assess the DNA methylation status of KCNJ10 in an enriched population of astrocytes and correlate DNA methylation changes of the promoter with transcriptional activity. This is accomplished by first isolating an enriched population of cortical astrocytes from whole rodent brain via fax sorting. Next, DNA is isolated from the enriched population of astrocytes. Then, the DNA methylation status of KCNJ10 is assessed by using methylation-sensitive high-resolution MELT analysis, or MSHRMA. Finally, the KCNJ10 promoter is hypermethylated and a luciferase assay is utilized to correlate changes in DNA methylation of the promoter with transcriptional activity. Ultimately, MSHRMA and the luciferase assay are used to measure the DNA methylation status of KCNJ10 and correlate changes in the methylation of the promoter and transcriptional activity of the gene.
Demonstrating the procedure will be Sini, a postdoctoral fellow from my laboratory.
All animals were handled in accordance with the National Institutes of Health guidelines. The Animal Care and Use Committee at the University of Alabama at Birmingham approved animal use. All buffers and reagents were prepared according to the text protocol. After dissecting the cortices from the head of a euthanized animal according to the text protocol, cut or Dremel a hole in the top of a 50 milliliter conical tube to allow tubing to be fed into the tube. Then